Welcome everyone to the Water, Wetlands, and Watershed Seminar. It is Wednesday, March 23rd, and I'm going to hand over the introduction duties this time to Dr. Kathy Todd Brown. So Kathy, please.
Thank you.
Oh, wow, yeah.
In-person audience.
Welcome.
Excellent. So it's my pleasure today to introduce Schaeffer, who's a colleague of mine who's down from University of Tennessee. He is in the Department of Biosystems Engineering and Soil Science. He received his bachelor's degree from University of Utah, a master's degree from University of Arizona, a PhD from University of Arkansas, and did 2 postdocs at University of Santa Barbara and University of Utah. Dr. Schaeffer is a soil biogeochemist whose research interests includes soil ecology, biogeochemistry, sustainability, and climate change. He has over 55 peer-reviewed publications and is the elected chair of the Soil and Environmental Quality Divisions at SSSA, and is the director of Environmental Science, Soil Science undergraduate program in his department. So Sean has done extensive work looking at dryland and the interaction between water and biogeochemistry, particularly in drylands, but today he's going to talk to us about plastics and the fate transport of plastics in soil. So thank you so much for coming and talking with us, Sean.
All right, welcome. Yes, and I'll minimize you over here and do what you like, and you should be good to go.
All right, thank you, Kathy, for the introduction. I'm going to go ahead and take this off while I talk. I hope it's okay. So thank you for inviting me to come speak at the seminar. This is my first visit to the University of Florida campus. It's very beautiful. Not my first visit to Florida, though. Always enjoy coming down for a visit. Excited to talk to you in this department. I am in the Department of Biosystems Engineering and Soil Science. Bit of an bit of an odd marriage between soil scientists and engineers, but we make it work. And I want to talk about one of the results of that collaboration is the work that we've been doing on biodegradable plastics in agriculture. And I want to talk about that today, a lot of the research that we've been doing, but a lot of it also applies to non-biodegradable plastics just in the environment in general, and hopefully make the transition from soil systems to aquatic systems by the end of my seminar, because this is a wetland seminar, right? So if you need any convincing that plastics in the environment are a problem, I have a couple of introductory slides with very big numbers that show how much plastic is estimated to be produced annually, how much of the waste is generated from this plastic production, and all of these plastics are subject to fragmentation into progressively smaller and smaller particle sizes. And really what we're interested in looking at in terms of environmental contamination in our research are the micro and nano-sized particles. There's particles that we can't necessarily see, can't see nano-sized particles. Some micro-sized particles, maybe we can discern visually. But we really don't have a good idea in terrestrial environments what the impact of these micro and nanoplastic particles is. Not is going to be, it is, it's happening right now. Most of the research on micro and nanoparticle size plastics has come from aquatic systems, namely because there was a lot of popular press, and if you've heard about the Texas-sized islands of plastic trash in the Pacific. There's a slightly smaller one in the Atlantic. That spurred a lot of this research, and like most things, as a biogeochemist, like most things, the aquatic biogeochemists are often ahead of us in soils because it's a little more difficult to do things in soils compared to aquatic systems. So you can sieve through 50 liters of seawater relatively quickly to isolate plastic particles. That would be what, like the equivalent of 75 to 80 kilograms of soil. And that's just, it's not even feasible to think about doing that. So soils aren't the same as aquatic systems, we have to worry about a lot of interactions with soil mineral particles. We have to worry about feedback effects on other soil properties, like bulk density, so the structure of soils, the water holding capacity of soils. And there are slightly different degradation pathways in soil systems as compared to aquatic systems. We have both physical and biological breakdown, just like you would have in an aquatic system. But we have different organisms. You know, we have probably a lot more autotrophs, a huge variety of macro and invertebrates and microorganisms in soil. My focus has mostly been on microorganisms, particularly bacteria in soil. So these are just a couple of figures from several reviews on the topic of plastic particles in soil, and the end result is the same as for aquatic systems. We're interested in the impacts on human health and food security. Maybe different from aquatic systems, we're also interested in crop production and soil fertility. And these slides are probably outdated because there's a new review coming out practically every week. In fact, we should have one coming out in the next couple of months. But today I really want to talk about research that we've been doing at University of Tennessee in collaboration with partners in Washington State, so part of Washington State University, where we were looking at agricultural biodegradable plastics. So if you're not familiar with agricultural plastic films, there's a couple pictures of how plastic films are used in agriculture. These are mostly polyethylene, non-biodegradable films that are laid down on the soil surface, and crops are planted directly into these plastic films. And they're used for a variety of purposes. They're used to maybe retain water, heat up the soil at a certain time of year, cool down the soil at a certain time of year, suppress weeds and pathogens. They have a variety of different uses. They come in lots of different colors, from black, white, clear, green, brown, all kinds of colors. So they are used extensively around the globe. And if you want to go to a place where they're being used even more extensively than here, it's China. China's the #1 user of plastic films in the world, and only growing. They have about 10% of their arable land underneath plastic right now. It's just a huge volume. So these plastic mulch films, like I said, are made from polyethylene. The feedstock comes from fossil fuels. The films are not biodegradable. They don't break down in the environment, which is their advantage. They're strong, they're lightweight, they're cheap to make. Their drawback is they're costly to dispose of. They cannot be recycled because of the amount of soil that clings to the films. And farmers don't leave them on the ground. They have to be physically pulled up. You can see a machine laying it down, but often they're removed by hand. That's a significant upfront labor cost in disposal. So there's an impetus to shift to the use of biodegradable films in agriculture to be a more sustainable alternative to something like polyethylene. So these films are 100% biodegradable by the ASTM standards for biodegradability. They meet that. The feedstock can be a blend of either bio-based or non-bio-based. So you can have petroleum-derived plastic films that are 100% biodegradable, but the feedstock doesn't come from a sustainable source necessarily. They cost about twice as much to manufacture as polyethylene films. There are issues with their strength and durability in the field relative to polyethylene because they need to be strong enough to function like polyethylene, yet thin enough to break down quickly in the environment. So there's that high upfront cost. However, there is supposed to be no disposal cost because Instead of removing them by hand at the end of the growing season, you simply till them into the soil and they break down naturally. Now, the ASTM standard for biodegradability says 90% of the material needs to be converted to carbon dioxide within 2 years, which does happen in the lab. I can tell you from 5 years of research in different environments, it absolutely does not happen in the field. It takes a lot longer, 2 years for these things to break down in the field. And it is very dependent upon climate, both temperature and water availability. So these have unpredictable breakdown in the field. Other barriers to adoption, why farmers don't use these, is because they look just like polyethylene. When you till it into the soil, you get big chunks of plastic. They look like non-biodegradable plastic. Just laying on your field, it looks dirty. The wind picks them up, blows them into fences, and people complain. So it's been a bit of a rough go trying to increase adoption of these materials. If you're interested in what the chemical structure of these compounds look like, on the top left is the polyethylene monomer. So this is the non-biodegradable material. And we've done research on all these other materials. These are all commercially available, except for number 3, which was an experimental blend that we developed for testing at the University of Tennessee. And most of these biodegradable materials consist of a polymer blend. And the 2 that I'm going to focus on, and that you'll see in this figure, are either a mix of polylactic acid and polyhydroxyalkylamate. That's this number 3, experimental PLA, PHA blend. And then the other is a starch PBAT mixture. So starch and PBAT is polybutylene adipate. These are all these biodegradable polymers are polyesters. So they degrade much more easily in the environment compared to polyethylene. Like I said, polyethylene is non-biodegradable, but there actually are bacteria that can use polyethylene as an energy source. Just recently found this out, but they do it extremely slowly. So there, in no way would you consider it to be really biodegradable in the conventional sense. Whereas these other blends, these blends of polyesters are much more readily degradable. The reason we use a mix of polymers is we try to mix a polymer that doesn't break down quickly, like polylactic acid. You mix it with a small amount of something that's more easily decomposable by microbes, like PHA, for example, or starch. And what that does is the more decomposable polymer sort of jumpstarts the biodegradation process and helps it happen more quickly. The reason we don't make it completely out of something like PHA is because it breaks down too quickly. It's not strong enough to perform like a polyethylene film. So there's a lot of work that goes into getting the right blend of these polymers. The research that I and others have been doing, all of the research I'm going to talk about comes from a large multi-investigator project, a specialty crops research initiative funded by the Department of Agriculture. And my part was to look at the fate of biodegradable mulches. I'm a biogeochemist, so I was interested in what happens to the carbon that comes from these biodegradable mulches, and I'll talk about that in a minute. But what this figure shows is the 2 main pathways for degradation of polymer mulch components are biotic and abiotic. So you have microbial populations that generate enzymes that depolymerize these materials and break them down. Those microbial populations are subject to changes in climatic conditions. They're sensitive to oxygen content, water content, pH, and temperature in the soil. But you also have abiotic agents, like particularly UV radiation. When these materials on the surface, they are subject to photodegradation. from UV wavelengths. You also have physical, mechanical, and thermal stress that leads to the breakdown of these materials. And the eventual fate that we hope for all of the carbon in these polymers is that you'll be converted to either carbon dioxide, released to the atmosphere, carbon assimilated into microbial biomass as microbes grow and reproduce. And then once they die, perhaps that necromass becomes part of the bulk soil organic matter pool. And that's important for things like soil health. We can build organic carbon content in the soil, it becomes healthier and generally more productive for agricultural purposes, also good for storing carbon in soil and keeping it out of the atmosphere. So like I said, we had a large 5-year project where we did a set of field experiments, both in East Tennessee and completely replicated in Washington State. I'm only going to be showing results from the East Tennessee plots for this presentation, but this was our field site next to the Tennessee River. This is like, it's about a 5-minute drive from campus. So not a bad place to work. We tested, like I said, 3 commercial biodegradable mulches, 1 experimental mulch, compared that to controls where we had no mulch application, a traditional polyethylene mulch, and then also a cellulose or paper mulch, which breaks down very, very rapidly in southeastern climate. So today I'm only really going to show experimental results from this PLHA, PHA blend and this starch polyester, the starch PBAT blend, just in the interest of time. So I mentioned physical weathering of these materials on the surface. This is the first stage of the breakdown of these materials. They actually weather on the surface. And we've got several papers, this is just one of them, published on the first stages of physical weathering. And we can tell that plastic becomes more brittle and mechanically more brittle and measurably thinner as it's weathered on the surface. And that's mostly due to the effect of ultraviolet radiation and breaking down the polymer chains of these materials. And you can get here, I've shown some data, you can get I have data on cross-linking, but what happens is these polymers, if they're laid down in fibers, can actually start to form cross-links as they weather. It makes them a little more difficult to degrade. But there's also changes in the polymeric composition. That's what these data show. And so all this is for A and B. A is the PLA, PHA blend. Panel B is a PBAT starch blend, 2 different biodegradable mulches. And the bars show, nice University of Florida copy. The bars show the proportion of the 2 different polymers that make up that material. And you can see as they, over time, so from 0 months to 12 months, if you put both weathered, previously weathered and unweathered material just on the surface, what happens is over here, that PHA, the orange bar, and here starch, the same orange bar, panel B, those more easily degradable polymers, those tend to disappear first as these materials sit on the surface. So we can already see the chemical composition of these materials changing before it even makes it into the soil. And that's important because actually the surface weathering does help increase the rate of biodegradation once these materials are incorporated into the soil, even though there is some cross-linking that occurs. So just to give you the theoretical idea of what I'm talking about, we have to get from large particles, meter square sheets of plastic, down to nano-sized particles, down around the hundreds and thousands of micron in diameter. And generally what we think is occurring is you have physical effects, more important on large size fragments, and chemical interactions more important for smaller size particles. And this is driven by both, you can see both the physical and biological factors occurring here. This, hey, I can use this now. Right here in red, we did develop a method for visually isolating microplastic particles in this size range from 250 microns up to 2000 microns. And so this is a combination of particle size fractionation and then density fractionation. So first we separate particles based on their size class, and we have a whole bunch of organic, particulate organic matter in there from plant litter. We can use a density separation to separate the plastic material from that. Once we've isolated the plastics, we can use a microscope and image J software to actually measure the surface area of all the particles that we isolate. We already know the thickness and density, carbon content of the material, and then we can calculate the amount of carbon and the total amount of mulch that's left behind in the soil. So we did that for 2 years in this experiment. And you can see this is what we found in the table at the top. For 2 different biodegradable materials, we added about 7.3 grams of carbon per meter squared from mulch material. And then because we did it for 2 years, we just double that in the second column. So we know we've added 14.6 grams per meter squared carbon. And then when we measured the undecomposed residue in just that 250 to 2000 micron range, this is what we found, 1.2 grams or 0.8 grams, so about 1 gram of carbon per meter squared left after 2 years, but only in that very small size range. This doesn't include all of the plastic that's in the soil. But what we do see is we do see plastic residue accumulation over 2 years. So if you look at the bar graph in the bottom right, you can see we have on the left a no mulch and polyethylene treatment. So the no mulch got no mulch, polyethylenes were removed at the end of the growing season. So you don't see a lot of accumulation of plastic particles. But for all of the biodegradable materials, you do see a significant amount of accumulation. And so, you know, our ongoing research is, well, what happens if you're using this every year? And you would be if you were a grower. So, you know, we have to take this into account when we're looking at the loading of plastic particles in the soil in particular. And then obviously, where does that material go? Does some of it go into groundwater? Does it go into water? Things like that. So even though it's biodegradable, it will go away eventually, but it is accumulating in these soils. Me being a biogeochemist, I wanted to know something about the carbon cycle in the soil and how the addition of these plastics was affecting the carbon cycle. So we know we have inputs of carbon from both plants and from the mulch materials themselves. And once that material makes it into the soil, it is either stored in the soil as soil organic carbon or microbial biomass. And in my lab, we can partition out that soil organic carbon into many different fractions. I've shown here 7 different fractions of organic carbon that we looked at. And then we also looked at losses of carbon via CO2. And to make a long story short, there was really no consistent effect of biodegradable material on any of these carbon stocks. Some were high, some were low, some didn't change. It was different for every material. The only consistent results that we seemed to find were with particulate organic matter. So this includes both plant and plastic mulch. in the soil. And so we just did a straight particle size fractionation of these soils, and you can see for these soils, most of the mass of the soil, if you look in the bottom right, 56% of the mass is in the fine sand fraction. But actually, 56% of the total soil carbon is found in the silt and clay fraction. And this is very typical of what you find for particulate organic matter in soils. You tend to find most of it gets stored in finer fractions. So this is sort of an estimate of the long-term, this would be a slowly turning over pool, relatively slowly turning over pool of carbon. So here I have 3 panels that show coarse sand, fine sand, silt and clay fractions for all these materials, for all of these treatments, no mulch, polyethylene, cellulose film, and then 4 different mediums. And the only really consistent results we find are in the silt and clay fraction, where you get no change in carbon storage over, this was again over 2 years, no change in carbon storage in this fraction. For polyethylene, positive change for all of the biodegradable materials. And then just to make things difficult, the same amount of change in a control treatment. So the way we interpret this is that at least biodegradable materials don't have a negative impact on carbon storage in the soil, where polyethylene might actually reduce the amount of carbon storage over time. And that's not really surprising when we look at, in terms of a carbon budget, adding up all of the carbon atoms and where they're going. The actual amount of plastic-derived carbon that we're adding to the soil is very small compared to what is there already and compared to what is being added every year by the crop. So here's an example of all of the materials in our experiment. We know the amount of carbon, the percent carbon of each one of those materials. So we can calculate a theoretical amount of carbon that we're adding. So we calculated we're adding anywhere between, say, 6 and 28 grams of carbon per meter squared. It sounds like a lot. Well, most crop residue is in the range of 100 to 500 grams of carbon per meter squared per year. So it's about 2 orders of magnitude more being added by plants. And that means that this is actually, it's actually falling almost outside of our ability to detect it using available instruments. That amount of carbon being added to soil. You would have to use some isotopic tracer, carbon 13 or carbon 14, to really quantify this. If people have done this, it is still small. The impact is going to be indirect because the mulch films actually modulate soil moisture and temperature. That's the main impact that they have. But in terms of the degradation rate of plastic particles in the soil, we did look at the effect of temperature. It's going to be one of the overriding factors controlling degradation rates. So what we did was we did some incubations where we took different mulch materials, added them to cut them up, added them to soil in the lab, incubated them for 16 weeks at 3 different temperatures, and then looked at rates of biodegradation. And we measured and calculated biodegradation by the amount of CO2 that was released. And you can see when you look at the cumulative amount of carbon dioxide released over 16 weeks, that as you increase temperature, you go from 10 to 20 to 30 degrees C, you get increases in biodegradation. And you do get that. You actually look like you do get increases in degradation rates over and above what you see in control treatments. I'll show you some of that data in a couple slides. we did try to address this idea of temperature sensitivity. And one way to measure the temperature sensitivity of biodegradation is to calculate what is called the Q10. And Q10 just means, the definition of Q10 is the increase in the rate of a reaction, any reaction, with a 10-degree increase in temperature, which is why we did 10, 20, and 30 degrees C. Nice, makes the calculation easier, right? So we calculated the Q10 from 10 to 20 degrees, the Q10 from 20 to 30 degrees, and then the Q10 from that whole range, 10 to 30 degrees. And what we see is no effect of adding BDMs. That doesn't change the temperature sensitivity of the entire system. Because remember, not only is the biodegradable material breaking down, but so is the organic matter in the soil. So adding these materials, I mean, it's a good thing. It doesn't change the metabolic temperature sensitivity of the entire system. And we do see a slight increase in the overall Q10 as we go to a lower temperature. This is, again, very common to see in most ecosystems. At lower temperatures, decomposition tends to be more temperature sensitive. And if you're looking for a general value of Q10, 2 is about what we say. So we mean metabolic rate doubles with a 10-degree increase in temperature. Like you can see, we start to creep up above 2 as we decrease the temperature. We did a quick calculation. We had controls where we didn't add plastic mulch, which allowed us to get an idea of the percent biodegradation of these materials over 16 weeks. So we know how much carbon we added in terms of mulch carbon. We know how much carbon was in the soil organic matter to start with. And then we know how much was released as CO2 over the entire incubation. So we can do a rough calculation of percent biodegradation, and that's what you see in the red here for 2 different materials. What we saw was we had one material, this PLA-PHA blend, which was really only degradable at higher temperatures. It wasn't until we got around 30 degrees Celsius that we saw a big increase in the percent biodegradation. We saw really no biodegradation at 10 degrees Celsius, and then only 3% at 20 degrees C. Compare that to a starch PBAT blend, where you do get degradation happening at 10 degrees C, and then up to 15% at 30 degrees C. And so this highlights the fact that climate is going to be a big factor in how fast these materials break down in the environment, but also the polymer blend can significantly impact that. And if you're interested after the talk, I can explain why this is so much different than this. It's a kind of an interesting story, but it would probably take too long to explain right now. So given this unpredictable biodegradation in soil, can we say anything about the effect of these materials on soil quality? Does using these materials make our soil more able to store organic carbon? Does it make these soils more amenable to growing crops sustainably in the future? And the practical upshot is, again, the answers are all over the map. So this is just a meta-analysis of different studies that have measured different aspects of soil health. I'm not going to go into them, but they're all listed in the table. And you can see that they've looked at different materials, they've used different parameters, but compared to bare ground, you do tend to see some increase in these soil health or soil quality indexes compared to bare ground, but then compared to other BDMs or compared to polyethylene mulches, you often see no effect, and in only one case you see some increase. So again, what we've relied on, and I don't think our study, no, I don't see Henry's paper in here. But what we ended up concluding was that you can say that biodegradable emulsions don't decrease soil health, and that's a good thing because sometimes polyethylene films can lead to a decrease in soil health because they increase respiration rates. They tend to suppress microbial biomass growth, which negatively impacts soil health. So at least mediums are neutral in terms of soil health. And now our ongoing work, what we're doing now, our SCRI project has ended, but we've gotten funding from USDA to continue work looking at the fate and transport of these plastics, to actually see where they're going in the environment. So we're interested in the accumulation rate of these particles in the soil compared to the biodegradation rate, to see if we can actually get at some plastic mass balance for the system. And then looking at, as part of that mass balance, looking at vertical and lateral transport in soil. And also, because we didn't look at it before, ecotoxicity to soil organisms. Because even though these particles are biodegradable, they may have physical impacts on soil organisms that we haven't yet thought about. And they are going to be accumulating. We showed how they're going to be accumulating in soil if we continue to use them. So I had one of my graduate students, Alexis Gilmore, do a quick back of the envelope calculation to look at, if we assume that, say, the ASTM standard says that 90% gets biodegraded over 2 years, sorry. That remaining 10% over 2 years is going to accumulate. So, just do a quick spreadsheet calculation. You can see just linear accumulation of material over time. I had her try to fit some just rough degradation rates in the soil to that, to see if we could get at some, just to see if the accumulation rate maybe plateaued at some point. And I already know that these calculations are wrong. So we can't get. I already know it's not accurate. We've already amended them. But what we are seeing is that yes, you can reach a plateau of accumulation after a certain amount of years. And if you plug in different degradation rates, you can change the height of that plateau and how far out it is. And we know from the work that Alexis has done now, looking at soils deeper than the tillage depth. So we know that plastic particles are actually making their way down past where we have that tillage disturbance. So we know there's some mass transport of particles below 30 centimeters. We found plastic particles down to 60 centimeters in our field plots after 3 years. We don't necessarily know the mechanism for that, whether it's physical due to water movement, biological due to soil fauna, probably a combination of both, but it is happening. We're also working on quantification of non-visible particles. Like I said, we've so far only focused on the small visual range, but we can use proton NMR spectroscopy to look at those specific polymers. And this is also from Alexis's work. We can identify PLA, PHA, starch, and PBAT in a soil. And isolate it in most cases from the soil organic matter that's there. And so we're working on actually making this a quantitative method. So that will allow us to calculate the total amount of those polymers in a soil. It won't tell us anything about the particle size distribution, but at least we can get an idea of the total amount of the material in the soil. And just like the reviews, there's a new paper coming out practically every week on new methods for quantifying and characterizing plastic in the soil. We've also seen evidence using this NMR technique of lateral transport. So we know where on the fields we've added these polymers. We can go to areas that are outside. Like this is an area that's actually 30 meters away from where we did this experiment. We can find evidence of these polymers in the soil. So we know they're being transported laterally. That transport is either surface erosion or wind. It's moving these materials. It's farther than the machinery would move it. And this brings us to the work that we're just now starting. This is a project that's beginning in May at the University of Tennessee with some of our colleagues at Washington State. We're going to be trying to, this is the link to the water here, trying to look at the terrestrial aquatic interface. Can we see movement of agricultural plastics down into the vadose zone or hyperic zone of streams to see if it's being accumulating in groundwater or being transported in surface water. Luckily, our field sites are right next to some surface water, so we're going to be looking at surface erosion. And this work is being funded internally by the University of Tennessee through a strategic planning and research initiative, or SPRINT program. So the PIs are Doug Hayes and myself. Doug is a biopolymer engineer in our department. I'm handling the biogeochemistry. And we were funded to put together a multi-institution transdisciplinary team with some nice buzzwords, including hydrologists, agronomists, extension specialists, you name it, to look at this terrestrial aquatic interface. Starting in a couple of months, we're going to be using some flumes and weirs that are already constructed out of this at the same site that I was talking about to look at this erosion and lateral transport of these materials. We have other institutional partners that include Washington State University, University of Nebraska. I believe there might even be some interest from University of Florida in this, and also Cal Polytechnic University in San Luis Obispo. So if you are interested in hearing more about this project or potentially participating, we're just now getting ramped up, please feel free to contact me. I'm happy to explain this in more detail. And my last slide is just to acknowledge our funding for all the data that I actually showed you was from our Specialty Crops Research Initiative. This was a large 20, 25 PIs over 5 years. So just wrapped that up in 2019. And so all the data I showed came from that one project. So yes, thank you very much for inviting me. I'm happy to answer any questions. YouTube, Zoom.
All right, well, we don't have any questions in YouTube yet, but so YouTubers, please drop questions for Dr. Schaeffer there, and I'll check it on the Zoom. Same goes for you folks there, Madeline, Amanda, and Lena, etc. So I'll just kick us off. So not knowing too much about microplastics, except hearing about them all the time. You mentioned you're going to do some in the next round, some ecotox work, but in general, do we know are the effects on organisms differential between sort of classic microplastics versus sort of these biodegradable microplastics? Is there an expected mechanistic difference in their toxicity or their health effects on organisms?
So some of the impacts, it wouldn't be strictly an ecotoxicity impact, but just the physical interaction of these organisms with these particles can impact the organisms. So these particles can stick to the surfaces of cells, can stick to the outsides of organisms, or they can ingest them like earthworms. So even though the particles may be not toxic in and of themselves, they have that physical impact, but there are also other things in those plastic films that are not generally biodegradable. So in particular, you have the colorants that are used. So if you have a black film, The colorant usually comes from carbon black, which is basically carbon nanoparticles. If it's a white plastic film, the colorant here in the US is titanium dioxide, which is known to be directly toxic to bacteria. If you've ever bought antibacterial mats or antibacterial sandals, you know, smell nice.
Yeah.
They're impregnated with titanium dioxide or silver nanoparticles in many cases. So we know those elements are in, you know, small amounts, even in these biodegradable products. So we're interested in both of those effects.
And that would be, I mean, so that would, for regular plastics that are colored, would have sort of the same thing.
Yes,
So it's not
same
just-
thing with regular plastics too.
So I had a question, also coming from a point of knowing very little about microplastics. So I noticed that you developed this methodology to identify or quantify the particles within a very specific range. And it sounded like you spent a lot of work
It was.
developing that protocol. Was there a reason for why you picked that range in your quantification?
So this was work done by a master's student of mine, and that range was the most easy to isolate based on all of her experimentation. She spent a lot of work trying to expand that range. Below that range, you can't reliably identify plastic particles through microscopy.
Gotcha.
And then above that range, there is a lot more particulate organic matter coming from plants. It makes it much harder to do the, particularly the density fractionation. So that size range sort of represents the sweet spot of where we get the most easily identifiable plastic particles.
Gotcha.
That's why we picked it.
Good reason.
You know what it reminds me of? Like, you know, in the aquatic environment, when you're trying to count algal cells and you need like microscopy and an expert, and you're doing it by, you know, by the physical structure, the physiology of the cell, and it takes hours and hours. And now there are these cameras where you kind of, these cytocams, and you pass the sample through, and based on image libraries, you can sort of partition, like, you know, species A, species B, et cetera. And it seems like, are there potentially differences in the sort of the way, because you're looking at particles, right?
Yeah.
So the particle is a thing, right? And
Mhm.
it has edges, it has a shape, it has maybe some sort of characteristic shapes that could be learned, where you can count particles in an automated way that expand your capacity to go lower or higher.
I think we absolutely could for these plastic films because they have a very restricted shape. They're plastic sheets, basically. So yeah, they do have a particular shape relative to other things. I think that's absolutely possible. What people run into with just plastic particles in general is they're much harder to pick out based on their shape because you get all kinds of different shapes.
Yeah.
And all kinds of different colors. If it's pink or purple, then it's easy
Right.
to isolate. So yeah, I think that absolutely could work for what you're doing.
Well, maybe it's easier just to make a pink one for the experiments, right? I mean, that's a great idea.
Well,
Simpler.
one of the things we're trying to do is really scale up these estimates. So can we look at a larger area? And what we're settling on is it's incredibly time and cost intensive to sift through soil to look at these particles. And so in order to expand the spatial scale at which we can look at these these particles, we're kind of shifting towards large visible particles. So ones that are larger than 2 millimeters in diameter, or particles that just sit on the surface of the soil, because there we could actually use spectroscopy and remote sensing, believe it or not, to identify plastic particle distribution across a landscape. So that's probably where we're going to go for upscaling, but we'll still have to do some of this, plot scale characterization at some point. And it'll be a combination of what use the visual techniques, but also some chemical techniques like FTIR or proton NMR, depending upon the polymer.
All right, well, seeing no takers from YouTube, there are people on there, but they don't
I
have
have a
questions.
question. I don't know if you were.
Yeah, please go ahead.
I have two quick questions. When you said accumulation and the biodegradable agricultural plastics accumulate faster than we expect, what are you talking about accumulation? Are you like saying like the carbon accumulates faster from the materials? Or what exactly is it?
Both. The particles are accumulating in the soil. The particles have, about 40 to 50% of the mass of those particles is carbon.
Okay.
And so they're biodegradable, meaning that eventually they will be converted into something else,
Yeah.
biomass or CO2 or soil organic matter. But they don't break down. They don't break down right away, and it takes more. If it takes more than a year for them to break down, which it does, if you're adding more plastic every year, even though it's biodegradable, it's going to start to build up.
Okay, so do they need to add more? Do you need to add more if it still hasn't? Does like the use of the biodegradable plastic like lessen over that year, that span of a year that you have to keep adding more?
Well, so from a farmer's perspective, it's only useful when it's a plastic film on the surface of the soil, right? They're using it to cover the soil.
But you can't place it, plant, harvest. You can't till, so you have to remove it every year. I guess that's the.
If it's polyethylene, yes, you have to remove it every year. If it's biodegradable, you can actually mix it back into the soil.
But you can't.
But then it's not a plastic film anymore. It's little bits.
But you can't do the choice, like,
No.
That's what I was wondering, so you can get less, usage out of it, like more usage out of
Even a polyethylene film doesn't get reused. Those are almost always single use.
So you mentioned that one of the effects of this film was to increase the temperature, which increases the K rate. And then another effect is that you've got this sort of 90% reduction per year for each of the applications of these. More, we'll be generous. We'll say it's 90. Well, actually, let's not be generous. Let's say it's only 50%. So like you only lose half every year. Has anyone looked at sort of the carbon balance budget of these fields, and maybe not paying too much attention on whether it's locked up in the plastic mulch or if it's actually in like traditional SOC, and thought about the trade-offs between that change in the soil temperature that you're getting with the mulch application versus the shift in the recalcitrancy of the carbons in the soil?
For biodegradable films, I don't know if anybody's done that specific calculation, but they have done for both biodegradable and non-biodegradable films, is you can look at the difference in net primary productivity, NPP, of the crop. So we can calculate the rate of input change per year. And typically, you're using a mulch because you want to increase NPP.
Right.
That increases your yield. There's really no other reason to do it. Well, unless it's a pathogen control. So it would be interesting to do that calculation, although my sense is, as we looked at, there's a more than 2 order of magnitude difference between that NPP input and that direct plastic addition of the mulches. So my sense is it'll take a while for you to see that direct application of mulch derived carbon. It's probably more important to focus on how that mulch is impacting the plant input into the soil. And that balance can be positive or negative when you're using mulch. Because as you say, you're heating up the soil, you're increasing plant growth, but you're also increasing decomposition. And so the net storage may actually go down for in some cases when they're using the plastic film.
I have a quick question.
Yeah.
You mentioned that there's, for biodegradable, the polymers, it's a, you mix the one that is easy to degrade and one that is hard to degrade, so it can last longer.
Yeah.
I don't know if you guys ever like, as you're doing your research, you find like, what is the most optimal percentage of each polymer? To make
Yeah,
it,
it
like, last longer, but still like, because you, like you said, you don't want it to degrade too fast. You know, that might help it like not be accumulate, but.
Yes, and it depends upon what polymers you're using, what the optimal blend is. Although I will say that all of the commercial products, that what they consider an optimal blend isn't based on biodegradability. It's based on performance in the field. Is it strong enough and does it last long enough to perform the functions that the farmer needs it to perform? So biodegradability is actually a secondary consideration when looking at these polymer blends. So that's a very good question. Maybe biodegradability should be
It
the
should be
primary
one,
concern.
so that way they can accumulate it.
All right, well, let's do one more round of applause, Dr. Schaeffer. Thank you very much for joining us today. All next week for the next seminar, and until then, have a great week. All right.